Hi everyone, we're just waiting for a handful more people to come in from the keynote lecture, so just going to give it a few more seconds for people to find their seats. So a very warm welcome everyone to our Bacon-sponsored symposium on neural mechanisms of novel mental health interventions. I'm Charlotte Ray and I'm co-chairing today's session with my colleague Chatrin Suksasilp, who's down here at the front. And I'm standing in today as co-chair for Professor Sarah Garfinkel, who is the incoming president of the British Association for Cognitive Neuroscience. Sarah's been called away today, so I'm here deputising. But before we get going with the science, I just wanted to say a few words on Sarah's behalf. So Bacon, the British Association for Cognitive Neuroscience, is the society for British cognitive neuroscientists, numbering hundreds of members and supporting one of the largest national communities of cognitive neuroscientists in the world. Our next meeting is coming up soon in a few months' time on the 11th to 12th of September. That will be in Edinburgh, so do pop that in your diaries. We'd love to see you there. There'll be a keynote lecture from Tim Behrens and a number of fantastic symposiums on a wide range of topics, from the self to visual processes from infancy to adulthood. So our Bacon-sponsored symposium today is aiming to showcase ways in which we can use cognitive neuroscience techniques to understand mental health and hopefully how we can improve it. We're going to be following the traditional BNA symposium format of 4 talks, each about 20 minutes long, with then 5 minutes of Q&A for each speaker. So as you're listening to the talks, do have a think about questions that you would like to put to speakers, and we particularly encourage any early career researchers to ask questions. We'd love to hear from you. So I'm now going to hand over to Chatrin for the first talk. So Chatrin's going to tell us about really exciting approaches using computational psychiatry to understand the mechanisms of intervening for anxiety. Over to you, Chatrin.
Hello, thank you all again for coming to our symposium, and it's a real delight to be able to speak to you. So I'm a PhD student at the UCL Institute of Cognitive Neuroscience, and my PhD project focuses on the role of brain-body pathways in mental health disorders. And today I'll present on some computational modeling work that I've done on anxiety and interoception. So a quick background to this line of research. The brain is constantly processing signals pertaining to physiological events in the body, and we refer to these processes as interoception. So, for instance, the brain shows a widespread electrophysiological response to each and every heartbeat, which is signaled via arterial baroreceptors through the brainstem. And the respiration cycle also modulates brain activity, this time through sensory neurons in the nose, having a widespread effect on neural oscillations. At the same time, these physiological cycles appear to impact our cognition and emotion on a moment-to-moment basis in domains such as memory, fear, and pain processing. And obviously, mental health conditions very commonly feature somatic or bodily symptoms, such as palpitations in anxiety or lethargy in depression and schizophrenia. And one large study actually estimated that clinical anxiety comes with, on average, 5 different bodily symptoms. And so, given that these brain-body mechanisms appear relevant to mental health, it's interesting to ask if we can improve mental health symptoms via these bodily or interoceptive mechanisms. And I've been lucky enough to work with existing data from my supervisor, Professor Sarah Garfinkel's lab, suggesting that this is possible. Specifically, training people to perceive their heartbeats more accurately appears to reduce anxiety in both autistic adults and neurotypical adults. And this training is fairly straightforward. Participants' heartbeats in a cognitive lab are continuously measured using an electrocardiogram or a pulse oximeter on a finger clip. And in each trial of this training, participants are presented with auditory stimuli, which are tones that are played at the same rate as their heart rate. However, the tones can either be in phase with their heartbeats or out of phase. And we called these conditions in sync and out of sync. And so after hearing these tones, participants give a perceptual judgment of whether the tones were in sync or out of sync with their felt heartbeat sensations. And finally, they were given feedback as to whether they were correct or incorrect so that they could learn over time. However, little is known about how this cardiac training works to improve anxiety, and little is known about the predictors of treatment response. And so in my PhD project, I try to address these questions using computational modeling methods. So computational modeling tries to understand cognition and brain function in terms of algorithms, doing so by analyzing data from behavioral experimental tasks, going beyond just summary statistics of, say, accuracy or reaction times, and trying to delineate the underlying mechanisms that generated the task behavior. And it does so in terms of algorithms that describe how agents can optimally solve problems of decision-making or inference. And so this tends to be using models of reinforcement learning and Bayesian inference. And this approach allows us to decompose behavior into computational parameters or phenotypes, which describe individual differences in terms of these algorithms. And the hope is that these phenotypes will give us better explanation of the mechanisms of action and better prediction of treatment outcome. And so with these data sets, I used a Bayesian inference framework to analyze behavior. So I'll first present our preprint, which was from a study in neurotypical adults without a mental health diagnosis, half of whom did the cardiac training task I just described over 8 training sessions, while the other half were a passive control group that did nothing. And both groups also did assessment versions of the task that did not have any feedback to produce snapshots of interoception over the course of training. And so to elaborate on the mechanisms of how this worked, I implemented a partially observable Markov decision process model of the task, which looks like this, which is a bit intimidating, but I'll describe it on a more intuitive level. Our model assumes that cardiac perception is a process of Bayesian inference, meaning that on each trial, the participant combines their top-down prior beliefs with bottom-up sensory signals to infer a posterior belief about whether the tones were in sync with their heartbeats or not. And what the participant perceives on each trial corresponds to this posterior belief. So a key parameter in this model is the precision weighting assigned to cardiac signals, which controls the extent to which veridical sensory signals that the participant receives influences what they actually perceive. And this is where we suggest learning happens, in that participants learn to improve the mapping between veridical sensory signals and what they infer on each trial. We also posited that the incoming signal itself perhaps wasn't perfect, and this was represented by a lower level signal precision at the bottom here, which we assume to be a static individual difference. And the final parameter was a bias in prior beliefs, which could be interpreted quite simply as a response bias. So the model that I just described is the one that produced the best fit to the empirical data, but we did compare it against 12 other models, each of which represented slightly different hypotheses about which mechanisms are present or absent, asking if we really needed to include things like precisions, prior bias, or even learning at all to explain the data. And so the model that best reproduces the data represents our best understanding of the mechanisms at play, at least within the set of models that we tried. And then from this best fitting model, we can derive estimates of each parameter for each participant to use as latent variables of individual differences. So onto the results. Firstly, does the intervention actually reduce anxiety? It appears so, at least for trait anxiety on the left here. That said, this particular data set is limited by the fact that the control group is passive, so we can't rule out placebo effect. Secondly, based on the computational model that best fit the data, an important conclusion is that interoceptive learning involves increasing the precision weighting of cardiac signals rather than, say, learning to calibrate your prior biases. And secondly, that a lower level component representing the quality of the signal itself moderates the rate of learning. And this is quite interesting from a Bayesian or predictive processing perspective, in that if you want to learn about a signal, the quality of the signal determines how fast you can learn. And in the sample, that specific parameter was correlated with participants' mean heart rate across the training trials, suggesting that this signal precision parameter could represent something about the quality of the cardiovascular signal. And finally, do our computational parameters offer an explanation for individual differences in anxiety reduction? Yes, it appears so. Specifically, gains in interoceptive precision weighting, so the degree to which you learn to improve your precision, is related to greater reductions in trait anxiety. And this is controlling for age, sex, and baseline anxiety. On the other hand, the prior bias parameter was related to state anxiety reduction. And these effects are particularly interesting because the measures we conventionally use for interoception tasks could not explain treatment response. Okay, now moving on to some more recent efforts that I did with the data set from autistic adults, which was a randomized control trial with an active control, which found a significant reduction in trait anxiety in the intervention group, but not the control group, where we also have unpublished data indicating that the trait anxiety reduction persists even 1 year after the trial. As in the study I just described, the intervention appears to improve only trait anxiety, but not state anxiety. Yeah. And so for the computational modeling, I expanded the range of models that we considered quite considerably. Notably, I asked if this lower level signal precision, which we previously assumed to be a static individual difference, could instead evolve over time. And this would affect both the accuracy of posterior beliefs as well as the rate of learning of the precision weighting at the higher level. And it turns out the best fitting model for this dataset is one of the new ones. This model assumes that the signal precision increases between training sessions, which effectively is very simple. After every 40 trials, we just bump this variable up by a tiny amount, controlled by a new parameter that I called IP1 change. And to make things comparable across the autistic and neurotypical samples, I ran our new model comparison pipeline on the old data that we had from the neurotypical adults, and they are still best explained by the old model. And so what we can take away from this model comparison is that there's some evidence to suggest that there's slightly different mechanisms at play in the neurotypical sample versus the autistic sample. In that in both groups, there's learning to improve the interoceptive precision weighting rather than learning to calibrate a prior bias. There's also the effect of signal precision, a lower level component, but the difference appears to be whether this lower level component is static or dynamic, which affects both. So in the autistic group, there appears to be something happening in between training sessions that's allowing them to be both more accurate in the next session and learn at a faster rate in the next session. Although at this point, I should acknowledge that the weight of the evidence for the best model in the autistic group, quantified by the protected exceedance probability, is much, much lower in the autistic groups versus the neurotypical groups. Although ultimately, I think this is an issue that resolves itself with more data points and replication. So before I move on to results about anxiety, in computational modeling, one way of assessing a model's validity is to verify that certain quantities in the model correlate with things that they should, separately from data that was used to optimize the model. And in our case, the the data set in autistic adults includes confidence judgments that participants made after each trial, ranging from 0 to 10. And if our model postulates that a participant's response on each trial comes from their posterior belief, it stands to reason that the confidence rating should be correlated with how strong that posterior belief is. And so in our model, this is what a posterior belief looks like. It's just two probabilities of the likelihood, so the probability of giving an in-sync response or an out-of-sync response. And a participant may have a very strong posterior belief for in-sync. They may have a slightly weaker posterior belief in the same direction, or they could have completely unsure posterior beliefs. And well, these figures are a bit messy because every single trial is a data point, but we do see that confidence ratings are associated with the strength of posterior beliefs in the predicted direction. So higher confidence and stronger posterior beliefs go together. And this effect is independent of how confidence ratings generally increase over the course of trials, as one might expect. In terms of relationship with heart rate measures, it's a bit more complicated now. In the previous study, we saw that the lower level signal precision was significantly associated with heart rate, but now we've decomposed this lower level component into two parameters, the starting point of IP1 and then how much it changes over time. And so the interaction term between these two computational parameters is significantly associated with heart rate, but I find this much harder to interpret, and it makes me speculate that the story isn't so simple for cardiovascular correlates of the model. And finally, we arrive at explaining treatment response. Our new parameter, IP1 change, how much your signal precision at the lower level improves between sessions, is significantly associated with how much trait anxiety reduction participants derive from the intervention. So the greater the change in IP1, the more anxiety reduction derived. Intriguingly, at the higher level, learning to improve interoceptive precision weighting is associated with trait anxiety change in the opposite direction, and I'll get back to this. As for state anxiety reduction, if we use conventional measures, so the change in signal detection measures for interoception, we get significant explanatory effects for state anxiety, but not trait anxiety. And so it appears that, at least in this sample, the conventional approach and the computational approach offer complementary variables that explain treatment response. Now, returning to trait anxiety, it's interesting and puzzling to me that the association between the interoceptive precision weighting improvement is related to trait anxiety change in opposite directions in the neurotypical group versus the autistic adults group. And at face value, one might theorize that improving your capacity to consciously evaluate your cardiac sensations is somehow anxiolytic in neurotypical adults, but anxiogenic in autistic adults. But alternatively, it could just be because the models that best fit these two groups just work differently, in that we've decomposed, we now have two parameters that interact to represent improvement in the task. And perhaps that means that the contribution of one of them flips direction. And so I'm still trying to get to the bottom of this as I write up my thesis. So onto limitations. Firstly, we couldn't predict ahead of time who benefited most from the intervention using just baseline measures, which would have been a big advance towards deploying this intervention clinically. And secondly, when doing computational modeling, it's quite important to check that your model can reproduce participants' behavior on a trial-by-trial basis. And unfortunately, that's quite low in this study. But I think that's attributable to how notoriously difficult the task actually is. So if a participant's responses is truly random, then the best any model can do is reproduce 50% of them. And I think we're getting some of that here. And if we zoom out a bit and look at the overall accuracy, our models reproduce that quite well, actually. So I'm not too worried about this. Okay, so to wrap up, computational modeling, as we hoped, allows us to uncover the mechanisms at play in interoceptive learning, and it gives us an explanation of treatment response where conventional measures could not, in both of our data sets. And specifically, the changes over the time course of interoceptive learning can be best explained by an increase in the precision weighting assigned to interoceptive signals, and that these Bayesian precision mechanisms can explain individual differences in anxiety reduction. All right. I'd like to thank my supervision team, in particular Ryan Smith at the Laureate Institute, who taught me how to use this methodology from the ground up. And of course, welcome for funding my PhD. And thank you all for your attention.
Fantastic. Thank you, Chatrin. Super interesting application of methodology there. Any questions from the audience for Chatrin? There's a microphone coming. Sorry, just getting it switched on. It's coming to you.
Thank you. It was really clear and very engaging talk. I have a question. I actually have a question about the autistic results. Could it simply be that autistic people perceive sounds differently? Because in autistic children, it's clear that they don't habituate to repeated tones like normally developed children. So they take every single same tone as a new tone.
Right.
And they stay arousal throughout the session. So I don't know how long is your training. It could be that the case that make them that additional part that makes them.
I think that's an excellent point. And unfortunately, we don't have, for example, data from some kind of auditory stimulation task that might allow us to kind of tease apart this difference, right? So if we're saying that there could be a difference in auditory processing between neurotypical adults and autistic adults, hypothetically, I could put that into the model, but unfortunately we don't have any data about the auditory processing separately that I could base that on. But I think it could play a role. It's just that we don't have the data to look at it.
Thank you. Stephen.
Thank you. It's a very clear talk. Precision seems to be a very important part of your story here. Yeah. And if I remember correctly, you've got a relationship between precision and mean heart rate.
Yes.
And I was surprised that you wouldn't look at, for example, heart rate variability as being more likely to be involved in precision than the actual mean heart rate. I'm not sure what the mechanism would be for explaining why the mean heart rate is related to precision rather than variability in heart rate.
Yeah. So we were able to collect heart rate and heart rate variability, and I did look at both of them exploratorily, but only heart rate came up as a significant association with precision. So one thing I didn't cover actually was in the autistic adult group, heart rate variability was negatively associated with confidence. So it does appear that if the signal you're attending to kind of jumps back and forth, you are less confident, but it doesn't necessarily affect your task accuracy. Overall, though, I think that the story about cardiovascular correlates is more complex than just looking at heart rate, because the putative mechanism for how heart beats are transduced and sent to the brain is via stretch receptive neurons at the aorta, which, heart rate doesn't necessarily prove as, it's not necessarily a proxy measure for that specific neuron activation. So I would speculate that we should look at things like blood pressure on a beat-to-beat basis, or pulse wave analysis, or the distensibility of certain blood vessels instead. But it's just that that's what the data set had.
Just to add to that point, I would maybe think that it makes sense that heart rate's associated with precision, in that you have an inverse relationship between heart rate and stroke volume. So if you've got a greater stroke volume with each beat, you're going to have greater aortic baroreceptor firing. You might have greater somatosensory detection at the chest wall. That instinctively seems to make sense to me.
Okay, great, thank you.
Question at the back, thank you.
This is a remarkable work. Thank you very much. Could you please degranulate just a little bit in the interoception training you use in your work?
Sorry, could you just repeat the question?
Yeah, sure. Could you please degranulate just a little bit in the interoception or interoceptive training you use in your work, please?
Degranulate?
Explain a little bit.
Okay, of course. So the interoceptive training itself, you mean? Let me just go back to... So do you mean just going through this training procedure again? Yeah, sure. So we objectively measure participants' heart rate in a cognitive lab, and you can use an electrocardiogram for that, you can use a finger clip for that, and then the reading is sent to a computer that can then administer auditory tones at the same rate as your heartbeats, but they can either be exactly at the same time as your heartbeats or just slightly delayed compared to your heartbeats. And so this is a test of whether you can feel your heartbeat sensations that putatively is immune to just guessing your heart rate because you're detecting the delay between the tones and the heart rate. And then the training component is simply telling participants whether they were correct or incorrect on each trial so that they can learn over time. Is that better? Okay, thank you.
Great, thanks, Chatrin. So now time to move on to the next talk. Thanks, Chatrin.
Okay, yeah. Oh, yeah, yes. It's my pleasure to introduce Dr. Camilla Nord, who will tell us about translating insights from neuroscience into acute augmentations of psychological therapy.
Thank you, Chatrin, and also thank you for such a great talk. So Sarah Garfinkel asked me to speak about treatments today. So I want to tell you about one stream of work in my lab, which I think exemplifies how cognitive neuroscience could actually influence mental health treatments. Now, speak to an antipsychiatrist and they'll tell you that the worst thing about neuroscience is that it hasn't managed to influence mental health treatments whatsoever. That's an exaggeration. There are lots of really good examples, but it has some grain of truth in it. Despite major advances in the neural mechanisms of mental health conditions, we haven't really influenced pragmatic clinical practice in most settings for mental health. That may be because this forward translational pathway is obscured by a lack of back translational understanding. So, in short, we don't totally understand what we're doing and why it works. There are a number of ways the field has tried to answer this, and I'll briefly tell you about a couple of mine. So in one case, we looked at brain scans, fMRI, during effective emotional tasks before and after a course of common antidepressant medication. And in this group of people, they showed common changes in the bilateral amygdala during emotion processing after selective serotonin or selective noradrenaline reuptake inhibitor treatment. In contrast, after a course of psychological therapy, patients showed changes in the medial prefrontal cortex. So these are anatomically distinct areas. And actually, even if you really p-hack the data and you just dig and dig and dig and you lower that level of significance, you don't see anatomical overlap overall between psychological and pharmacological approaches. That's what this particular analysis showed. But even though these are anatomically distinct areas, we all know these are not really functionally distinct regions. In fact, in a subsequent analysis, I showed that both overlap with what Lisa Feldman-Barrett calls the affect network of the brain. So anatomically separate, but functionally interlinked. That's one perspective, but it doesn't really get us into the kind of nuts and bolts of what a treatment is doing. It just gives us a kind of, perhaps a very general picture. So in a different sort of approach, I and other researchers have used computational modeling to look at what the mechanisms of particular treatments might be doing. In this case, we randomized participants to practice an active ingredient across many psychological therapies called cognitive distancing. And in the group who did cognitive distancing, there were effects on various components of reinforcement learning. And interestingly, these were sometimes overlapping and sometimes distinct from what we saw in the effects of SSRIs. So again, an interesting picture of where the actual nuts and bolts of what you're doing in psychological therapy might interact with an antidepressant medication or other forms of treatment. The final example I'll give you, which is in a sort of proper randomized control trial, is a way of using an understanding emerging from human cognitive neuroscience, in this case the fact that there's underactive dorsolateral prefrontal cortex activation in major depression, in a therapeutic context. In this trial, I combined it with transcranial direct current stimulation, but actually more famously and often more effectively in big clinical trials, this is used with repetitive transcranial magnetic stimulation, RTMS. In this trial, I used a series of sessions of cognitive behavioral therapy capitalized on those CBT sessions, which we know are very DLPFC intensive. They're very difficult if any of you have ever done a CBT session. And we augmented DLPFC activity using a transcranial direct current stimulation montage directly before each of 8 sessions. And it's that last example that many years later I thought, actually, although that particular trial results were pretty mixed, the idea is something that could be applied across very many domains. This was something we ended up calling acute augmentations, that you can perhaps enhance the neurocognitive mechanisms of a treatment, once you know what they are, once you've done that back translation, to enhance either the sort of speed or the efficacy of psychological therapy. So I've shown here a couple different brain stimulation techniques that have been used, but actually there are other more general approaches. For example, a series of studies have done exercise before CBT or other psychological therapy sessions. Maybe even more famously, this time emerging from animal behavioral neuroscience, some types of therapy, like exposure therapy, can be augmented with pharmacological interventions. I've got the most famous one up there, D-cycloserine, that can be combined with exposure therapy following a huge number of very interesting animal studies showing its effects on learning. And finally, my colleague on this particular investigation, Tim Dalgleish, and many other clinical psychologists have become very interested in augmenting psychological components of therapy. And this sounds a little bit like doing it twice, but actually it's taking a component of psychological therapy, for example, the component of decentering in mindfulness therapy, saying this is the real thing that matters, this is the active ingredient, let's boost that in a trial to boost efficacy. So a number of trials have taken that approach as well. In all cases, what they have in common is they're taking what you know about a therapy, the cognitive or neural mechanisms, and boosting them before, during, or after a therapy session. So when I had this idea, I thought, let's see if it works overall. And we ran at the time a big transdiagnostic meta-analysis looking at all the studies that had ever combined this. It was actually kind of hard because I'd made up the name. It's not a perfect name. Other people have used lots of different terms to call this, so it took us quite a lot of sifting, which you can see here. So in the end, we ended up reading 14,000 abstracts, so I'm very grateful to my co-authors on this study. In the end, though, we had a really decent power to look transdiagnostically at the effects of acute augmentations. And in this study, we used the primary outcome of the original clinical trial. So each study had, say, one condition where exposure therapy was augmented with D-cycloserine, one condition where exposure therapy was augmented with a placebo, or same deal for brain stimulation, same deal for some kind of psychological augmentation. And then we used whatever their outcome was, whether that was an anxiety score, depression score, and so on. We did a typical random effects meta-analysis, and then I know BNA is all about early career researchers, so this slide is one that only early career researchers can read. I've zoomed in for you here. You can see overall there was a mild to moderate but pretty consistent effect of these augmentations on clinical outcomes. And we sort of fiddled around with this in a few different ways. So there was quite a lot of heterogeneity, which you can see even in my zoomed in version there, but maybe a little better in the zoomed out one there. There's quite a lot of heterogeneity. But if you exclude the outliers, the heterogeneity goes away, it's not significant, and the overall effect remains significant. Maybe more interestingly, and this was actually a reviewer comment that was annoying at first, but ended up being very interesting, we reanalyzed it without the primary outcomes that the original study authors had used, using something called high top dimensions, which is a transdiagnostic framework that looks at, a sort of replacement for a diagnostic overview. And in this case, we found 2 transdiagnostic dimensions that acute augmentations were particularly efficacious for: fear and distress. These are common across anxiety, depression-related disorders. I also subdivided them into these 3 categories that I kind of previewed a moment ago, which I called somatic, sort of an old-fashioned way of including brain stimulation and body-based therapies in the same category, but otherwise I just wouldn't have had enough power, pharmacological and psychological categories. And if you look at pharmacological therapies, they have an almost identical result to looking across everyone. And this is probably because this is the dominant approach to acute augmentation. So there's a mild to moderate effect, pretty consistent, relatively large number of studies showing that if you augment psychological therapy with a drug, that may be a useful approach. I should point out there's a subset of studies that have a very different effect size in this category, and these were studies you could call psychedelic, although no real psychedelic researcher would, because I included cannabis in here. But these studies have a massive effect size, and there are probably a number of reasons for that. One is that they're much harder to control. If you take a D-cycloserine pill, you don't know it. If you take LSD, boy, do you know it. In psychological augmentations, we actually saw a slightly smaller effect size, and this is because the majority of studies in this category were cognitive bias modification studies, augmenting therapies with CBM, which we now know after actually about a decade of very thorough investigations is not as consistently efficacious as one might have hoped, although it is in some cases. And then finally, actually in my group, one of the categories we're most interested in, this category of somatic augmentations, had a moderate effect size, but a smaller number of studies than the others. So this is in some ways a kind of newer approach to interventions, but I'll maybe give you some previews of some interventions along these lines that we're working on right now. So overall, I've told you, I think, about a category of interventions that neuroscience is primed to improve, which are these acute augmentations of psychological therapy. The reasons we can do it is because we have a much better back-translational understanding of what happens in psychological therapy, and thanks to behavioral neuroscience in animals and cognitive neuroscience in humans, we have an understanding of how to enhance those mechanisms. So I hope to see many more studies like this in the future, and I hope that kind of naming this maybe gives some people ideas of what they can do in this dimension. Here are all some ideas that people are looking at right now, or peripheral to looking at. So things like beta blockers, propranolol, exercise, ketamine. We've got a study that we're working on designing at the moment, augmenting behavioral activation therapy with ketamine, TMS, which I've already mentioned, and everyone's favorite, psilocybin. What now? So I did all of this, and then that was sort of like, it was almost like a side investigation, peripheral to everything else you're doing as a neuroscientist, because often when you run something like an RCT, that is like years of pain. Boy, it's so difficult, especially I was an early career researcher at the time. It ends up really delaying your career. And so what you need as well is some really kind of quick and interesting discovery science studies, which then if they work out, you can then move towards that clinical idea in the future. And so that's what we've been working on recently, and many of them have been inspired by patient-public interaction work we've done. This is a paper that Lydia Hickman in the audience and Gabriel Mackey have just had out in Lancet eClinical Medicine, where we worked with a group of patients to talk about the influence of bodily symptoms on mental health and the ways in which these kind of body-brain interactions matter to them, some of which were what Chatrin is already investigating, like cardiac signals, and some of which were actually very surprising to us. So things like gut signals are very important, and it's relatively under-investigated in interoception. And another thing that really came out that I'll point out in a second is the idea of the timing of bodily signals when sensations occur throughout the day. So we have begun looking at this in a few different ways. In one of our streams of work, we're interested in changing the signals of the gut, and this is based on some basic science work that Edwin Dallmeyer and I did a few years ago, finding that there's a causal role of gastric rhythm in disgust avoidance. And this particular study combined changing signals in the gut with a pharmacological intervention with a very short-term version of exposure therapy, looking at disgusting things. And this is something that's actually quite difficult to do clinically. So exposure therapy for disgust is known to be very, very challenging. And we think one prospect could be enhancing it by augmenting, actually dampening down, in this case, the signals from the gut. In a separate series of work, we've become interested in circadian rhythm, and this is work led by my brilliant postdoc, Sarah Mehrhoff. So she did initially basic science investigation, looking at the relationship between circadian rhythm and effort-based decision-making, which luckily you guys are all experts in, thanks to Masood's great plenary. So using a task much like that, we showed that circadian rhythm works in very similar ways to apathy and anhedonia in terms of blunting your decision to expend effort for reward. But fascinatingly, this blunting, which if you look overall, it just looks like people with late circadian rhythms, those lazy people who love sleeping in, they have this blunted decision to expend effort for reward, completely reverses on its head if you test them at the right time for them. So if you test people with late circadian rhythms in the evening, they actually look highly motivated, non-apathetic, not anhedonic at all, compared to people with early circadian rhythms. So this was an interesting demonstration that not just psychopathological syndromes, but also your chronotype and the time of day you're being tested affects this motivation and decision to expend effort. Now, how could this be applied clinically? Well, this is an idea I had, and then in a beautiful series of circumstances, I told this to someone, and I was visiting, giving a talk there the other day, and they said, oh, we've actually already done that, and it works. And this is where you can time psychological therapy with someone's chronotype, so delivering especially motivation-based interventions in the morning for people whose motivation is highest in the morning, and vice versa for people who are later in the day. Actually something that doesn't happen, you know, the IAPT service is sort of burdened enough as it is, but it would be a very easy boost for people to be able to engage with psychological therapy. And then the final target I'll mention, and I will say, because this session was organized by Sarah Garfinkel, it doesn't have interception in the name, but there's a thread of interception throughout. We've begun working on ways of augmenting interceptive processing as a mechanism of also improving established mental health interventions. And I'll give you two examples of that. Oh no, I'll give you three examples of that we're working on thanks to a mental health award. Which is that we are augmenting it using, as Chatran described, a psychological approach, interceptive training, using a virtual reality approach led by Hugo Critchley, where people can see physiological signals from the body and learn to regulate them. And then in my lab, using a brain stimulation approach, transcranial ultrasound stimulation. And all of this is based on, so if you see on the right, this is a transdiagnostic investigation that we led, showing a common disruption in interception processing in the mid-insula across psychiatric disorders. So that's the target that we are focusing on for our non-invasive brain stimulation approach. And then on the left, you can see in a very fun review paper that I wrote with Sarah a few years ago, the fact that even established interventions, things we use day in, day out, change signals all over the body and neural processes related to interception, meaning we may already have an arsenal of putative interceptive treatments that we could harness and really take advantage of for therapeutic improvement. So I'd like to thank all of you for your attention, and most especially my brilliant lab who I get to work with on this.
Thank you so much, Camilla, for a very engaging and interesting talk. We have a few minutes for questions, if there's some in the audience. I saw this hand go up here, please. Do you have a microphone going around anywhere?
Thank you, Camilla. Lovely talk. The gut interoceptive element is really interesting, particularly in the context of the brilliant talks we had yesterday on the microbiome. So is that something that you're following up? What about something like dietary interventions?
Yeah, I'm so glad you mentioned that, Amy. It's something I've been thinking about a lot and not something I managed to work in here. So we haven't worked on the microbiome yet, but I agree, actually, dietary interventions could hold particular promise because we've recently found that there's a relationship between metabolic health and that same effort-based decision-making process as is common across apathy, anhedonia, and circadian rhythm. So that implies that there may be kind of external features of the body, modifiable features, that then would also affect effort-based decision-making. Actually, you could argue that may be the mechanism by which exercise is such an effective intervention, but it also implies that maybe diet is one route in as well.
It was really wonderful talk. My question is, are you using augmentation like transmagnetic stimulation or something along with the CBT and stuff like that in the clinical practice, or has there been a trial to do that, if it's useful or?
Good question. Are you asking if people are already using it clinically? Yeah. The answer is absolutely yes in terms of repetitive transcranial magnetic stimulation. It's not usually formally combined with psychological therapy. I think that's sort of the point that I'm making, that this is often something people do like almost accidentally. So actually this is something Helen Mayberg says about her deep brain stimulation trials in depression when I spoke with her about this work, and she said, oh, we've never thought about it that way, but actually everyone we give DBS to, we also give psychological therapy to. So it's kind of a sneaky augmentation, even though that isn't the way it was designed. And actually same with the psychedelics, I should have really made this point. Those aren't usually described as augmentations, they just are usually applied as augmentations. So in that sense, yes.
Thank you.
Thank you, that was a fantastic talk. Thinking about that review paper which you published in 2023 of augmentations, what we have is a field where there seem to be lots of options coming up, various terminology which people are sort of using to apply to it. It seems as if things are moving quite quickly. I wonder if you'd see any advantage in revisiting that 2023 review in the form of a living systematic review.
Yeah, I think that's a great idea. It actually was a review that came back to life once when a reviewer said I had to redo it by the end of the process of getting it reviewed. So it's a review I've sort of done that process with once, and in fact it would be great to be able to have that iteratively as a living review. Yeah, thank you.
I think we have time for just one last question at the back there.
I was wondering if you would recommend to generally assess more biologically relevant information in human neuroscience independent of clinical research, like, for example, chronotype or gut movement or something like that?
I would probably advocate for that. I think there's been, actually it's sort of a resurgence of interest in the influence of bodily signals on neural processing recently. I think for a long time bodily signals were thought of almost as a nuisance, actually for fMRI analysis, literally as nuisance signals. But recently we have become very interested in the information those signals are conveying and the importance they might have for cognition, behavior, things outside of the clinical domain.
Great. Thank you so much, Camilla. And next up, we have Professor Stephen Jackson, who will tell us about developing wearable neuromodulation based upon peripheral nerve stimulation to suppress tics in tic disorder.
So thank you very much to the BACN for inviting me to talk on this. So I'm going to talk to you about some work we've done in developing a wearable device. How do I move this forward? So tic disorders, Tourette syndrome, for example, is a neurological condition of childhood onset. It's fairly prevalent. It's the most common form of movement disorder in children. It's linked to hyperexcitability and autodynamics in the cortical striatal thalamic cortical circuits involved in the generation of movement. It's characterized by these unwanted movements and vocalizations known as tics. Importantly, tics can be suppressed, but the majority of people who have tics will report that suppressing their tics becomes extremely uncomfortable. They have a building up of something called a premonitory urge, which experiences a strong urge for action, and these feelings go away once the tic is expressed, but they return. So just a few words about the type of treatments that are available for tic disorders. The first line of treatment is psychoeducation, which is just telling people what Tourette syndrome or tic disorders is, followed by behavioural therapy, which can be successful, but it's very difficult to access. So for many individuals who report that they're on a waiting list for behavioural therapy that can be 2 or 3 or 4, 5 years long. And it's great deal of geographical variability. So in London, you may be able to get access to behavioural therapy reasonably straightforwardly, but outside of London, for example, in the northwest or the northeast or something, it may be something that you just don't get access to. The next common way of treating the disorder would be with medication. This can be effective, but as you'll be aware, the treatment for tic disorder is to give antipsychotics, so it's off-label treatment, and this comes with a number of adverse effects. And obviously parents are fairly loath to put their children on antipsychotics, so it's not a very positive thing, medicating children. Another very effective treatment is deep brain stimulation. So this is proof positive that modulating the brain circuit thought to be involved in generating tics can be effective. And just to show you, this is a cartoon showing what deep brain stimulation looks like. So there's a control device implanted in the chest. The stimulator is planted deep in the brain. So typical targets for deep brain stimulation will be the globus pallidus internal segment, which Masoud talked about earlier in his talk, associated with motivation apathy, but also the generation of these tics, and also the thalamus, the intermediate nucleus of the thalamus. The thing I want to draw your attention to is this. This is the percentage of improvement. This is a meta-analysis that was done fairly recently. A meta-analysis comparing DBS against medication against behavioural therapy. And what you see is that the efficacy of medication and behavioural therapy is roughly about 20%, whereas it's close to 50% for DBS. Typically as well, the patients being treated with DBS are the ones who have the most severe tics. So modulating that brain circuit is effective, and it's effective for people with extreme tic severity. The problem is that DBS is not considered suitable for children and adolescents. So the majority of people who would need it can't get access to DBS. So some years ago, we talked to families and patients about what it was that they felt they needed, and they came up with the following challenge. They said that what was needed was a low-cost, safe and effective non-drug treatment that could be used by the individual to give them control over their tics, ideally outside of the home. So this sort of rules out a whole bunch of potential non-invasive brain stimulation approaches that we might think about. And that left us with thinking, well, maybe we can modulate the peripheral nervous system through something like median nerve stimulation to modulate those brain networks involved in the generation of tics. And in particular, we thought about the idea then that the generation of movements is linked to particular brain oscillations, so mu and beta burst oscillations, which are associated, which have to be desynchronized to allow movements. And we thought, well, perhaps if we could modulate those, we could decrease the probability of tics occurring. So we had to think about how we might do that. And we thought about the idea then that we could entrain those brain oscillations with some sort of external signal and use that to increase the power of those brain oscillations and therefore decrease tics. So I'll say a little bit about that. Firstly, just to show you something about the relationship between those brain oscillations and movement. On the left here, this is data from Peter Brown's group some time back. This is those mu and beta oscillations in Parkinson's disease. So we have the power in those oscillations. On this axis is the frequency band we're interested in. And in this orange colour, we have when the patients their DBS is turned off. And in this dark colour, we have when the DBS is turned on. And what you see is that with the DBS turned off, you see a big increase in the power of those oscillations in this frequency band. And of course, Parkinson's is a disorder associated with problems in initiating movement and slowness of movement. By contrast, this is data from Mari Hallgrave's work, which was presented in a poster earlier this meeting. This is the same sort of thing. This is the power spectral density of MEG data. Again, this is the frequency band we're looking at. And here we're comparing the frequency band for healthy control participants against a group of people with Tourette's. And you see that contrary to the Parkinson's that have increased power, there's a decrease in power in that frequency of people with Tourette's, which is of course associated with increased unwanted movements. So suggestion was, based on this sort of thinking, if we could increase the power here of these oscillations that are associated with suppressing movement, then perhaps we can suppress unwanted movements. And the way we did this was to give a external signal, and in case we used rhythmic median nerve stimulation. So we're giving stimulation in the mu band, so 10 hertz or 12 hertz stimulation, electrical stimulation to the median nerve. And the idea is in the orange colour here, we have the sort of intrinsic frequency of the person's oscillations, and then we give an external pacing signal at a particular frequency. And the idea is with repeated stimulation, you see an entrainment such that the intrinsic frequency comes to match the extrinsic frequency. And we're inducing an increase in phase synchrony, which is an increase in power, and we hope that this leads to a reduction in the urge to tick. And this is work again done by Mari. We looked at this using MEG. We put people in a scanner, and then we looked at giving rhythmic versus arrhythmic stimulation as a control condition. And this just simply shows, here is the effect for rhythmic stimulation. So we're giving, in this case, 12 hertz stimulation. The stimulation starts here at 1 second, for a second, and you see that there is this increase in power at the frequency of the stimulation, and there are harmonics to that, and this is absent in the case of arrhythmic stimulation. And the important part of the arrhythmic stimulation, it's the same number of pulses, the same intensity of pulses. The only difference is one is happening at 10 hertz and the other one happens at an average of 10 hertz, but it's arrhythmic. And this just shows that we see contralateral increases in power in the contralateral hemisphere in the case of the rhythmic stimulation that's absent in the case of the arrhythmic stimulation. So what this is telling us is we can use effectively rhythmic median nerve stimulation to induce some sort of entrainment of those brain oscillations. Is this effective clinically?
Yeah.
So this is a young man with very serious tic disorder. He scores 100 out of 100 on the clinical scale that's used. And what's happening here is he is asked to use his left hand to move a slider device to indicate how strong his urge to tick is. And at the moment, he's not received any stimulation, but at some point, we're going to start delivering rhythmic median nerve stimulation at 10 hertz to his wrist, his right wrist. I don't know how to move this on, actually. Ah, good. So at this point, Stimulation comes on. He's moving the slider device to illustrate that almost instantaneously the stimulation comes on, his urge to tick goes away. And this is something that people have reported time and time again, that this urge that they feel to constantly tick just seems to go away as soon as they start being stimulated. And he'll remain with very few ticks, and certainly all the florid ticks have gone away, until we turn the stimulator off, and then they come back almost instantly. But for the sake of brevity, we'll move on. You just take my word for it. When we turn it off, they come back. Based on this sort of work, we were keen to develop something that was clinically useful. So we, in partnership with a university spin-out company, we developed a prototype wearable device in which people could deliver this stimulation on demand. And the idea was that people may be happy to tick at home, for example, surrounded by their family, but they don't want to tick in a job interview, or while travelling on the train, or going to the cinema, or any other situations where they feel they wanted control over their ticks. So the idea of this is press a button and they would get median nerve stimulation at a particular power, and this will be calibrated for them, and then we would see a reduction in their ticks, hopefully. And this is, again, putting these people in the scanner while wearing this. We can see that when the stimulation's on, there's an increase in power in the same places while they're stimulated, but during sham stimulation, we see no effect. And sham stimulation is reducing the stimulation to 50% of threshold. So based on this work, we did a UK-wide placebo-controlled trial in which this was given to people for use at home. It involved 135 participants. They were split randomly or pseudo-randomly into 3 groups. One group received active stimulation, one group received sham stimulation, and the other group was on a wait list. After a month, they would get active stimulation. I won't talk about the follow-up. The outcome measures. During the course of the trial, we asked a subset of participants in each of the groups to do daily videos. So we would video them before they turn the stimulator on, during stimulation and afterwards, and then these were blind rated for the number of ticks that you could see in the videos by 2 people, and they were blind to not only which condition, but also whether it was before, during, or after stimulation. And we also used the gold standard clinical scale, the Yale Tick Severity Score, which is measuring how your ticks have been over the last week. So it's not a measure of how effective the tick treatment is while you're being stimulated. It's an offline effect. Overall, how have your ticks been over the last week? This just shows you. It's not going to work. Okay, so this is an example of what the videos look like. So this is one young lad. You can see his ticks here, and in a minute he's going to put his hand up and show you that he's turned the device on. Here we go. So he's going to press a button. Then he'll put his hand down, and we can edit this out so you don't see that bit when they're rating them. And then you can rate what his ticks look like before, after, and during stimulation. Okay, this is what the data looks like from that trial. So what we're looking at here is tick frequency per minute. So this is based on the analysis of the videos, and it's comparing the active group in red against the sham stimulation group in blue. And here we have pre-stimulation, groups don't differ. During stimulation, substantial reduction in both groups, but it's much greater in the people getting active stimulation. And after stimulation, again, you don't go back up to baseline immediately in both groups. And this is the number of ticks per minute. So this is a measure of how effective the stimulation is during the course of stimulation. But we also have data from the Yale, which is the reduction over the course of 4 weeks of this daily stimulation. And you see that for active stimulation, there is a large reduction in your tick severity, but there's no change no difference between the sham stimulation group and the weightless control group, suggesting that there's both an online and an offline effect of stimulation. So, one thing to point out is this is the number of people who were classified as responders. And we took as our measure of clinically significant response a 25 percentile change relative to baseline. And what we see is for active stimulation, there's close to 60% of people respond, whereas in sham situation, close to 70% are non-responders. But it's still not everyone. And there's an issue about who does and who doesn't respond and how that might be improved. So I've been asked to talk about mechanisms, so let's think about this. There are two potential things going on in this time. One is that during stimulation, there is entrainment of these brain oscillations, which may have effects on brain networks. And there's also the possibility of changes in brain plasticity. So first thing, this is some EEG data we've collected. This simply shows us that if we look at the period before stimulation, 1 second's worth of 10 Hertz stimulation and then post-stimulation, this is the intertrial coherence of the EEG data during stimulation. You see that both groups show increased coherence, which is what you'd expect, but that has ramifications because what you're doing is pulling together a group of neurons to fire consecutively in phase with one another, and that has potential implications, for example, in brain plasticity. This is the phase amplitude coupling. So this is the phase signal for the mu band, and this is the higher frequency gamma band coupling. And you see that again, during the period of stimulation, for example, from 0 to 1,000 milliseconds, okay, you see both an increase in phase power and also an increase in gamma amplitude. So we've got increased gamma amplitude coupling going on during the period of stimulation. This is some data. This is again work done by Mari and others in the lab. So the top slide here comes from a published study by a German group by Irene Nuner and colleagues. What they looked at is areas of the brain that activate in advance of a tic, which you might think, or they certainly thought as well, might be associated with this premonitory urge, then sort of neural correlates of a premonitory urge. And the important point is here that it involves the anterior cingulate and also bilaterally the insular cortex, which we've heard a bit about when we're talking about interoception and the possible role of the insular cortex in the sort of recognition of these internal bodily events. This is data from Mari showing with MEG, what happens in the period ahead of a tic. So similar to the fMRI study here, but using MEG, and it's showing increased power in the mid-insular, in the case of the mu frequency, ahead of a tic. And the important point here is if we look at these two slides, this is from an fMRI study where we simply give people median nerve stimulation, and then we look at which brain areas are activated by the median nerve stimulation. And the important point is you see the primary and secondary sensory motor cortex, somatosensory cortex, but importantly we also see the bilateral insular activations which overlap entirely with these areas that have been linked to premonitory urge. So although we're stimulating at the wrist, we're modulating brain networks that are linked to the generation of tics, including somatosensory cortex, primary and secondary, insular cortex, and potentially some other areas as well. And then lastly, this is a study, again run by Mari Hulgreave, and this is, again, looking at rhythmic 10 Hertz stimulation, periods of on and off stimulation, and this time we're using functional magnetic resonance spectroscopy to look at concentrations of GABA and glutamate in the somatosensory cortex during rhythmic and arrhythmic stimulation. So these are the areas for rhythmic and arrhythmic stimulation. And what I'm showing you here is the data for GABA. So this is the GABA-creatine ratio. And what you're seeing is that there is a modulation of GABA. So stimulation reduces GABA. When you turn stimulation off, GABA goes back up. Then it reduces again during stimulation, goes back up, etc. So we are seeing modulation of both GABA and glutamate as a result of the stimulation. So what this is telling us is that although we're stimulating at the wrist, we're able to have a clinically significant effect on people's behaviour. We're reducing their tics, we're reducing the urge to tic, and the underlying mechanisms for this are that we're potentially modulating brain networks within the brain that extend well beyond just the somatosensory cortex. And I'd like to particularly thank all of the people involved in the study.
Addressing an area of real unmet clinical need here. So we've got a question over here.
Very nice talk. I was just wondering if you see any effect on multiple such stimulations. I mean, kind of an habituation kind of an effect. So do you see that you need to increase the current amplitude or something to achieve the same amount of response in terms of tic avoidance?
So amplitude is calibrated to each individual. So a bit like most brain stimulation, we actually titrate the stimulation intensity to the threshold for that individual. So
sure.
Yeah.
But does it also change longitudinally for a given subject?
So does it habituate? Yes. The evidence that we've got so far says no. And the thing is as well that we don't give continuous stimulation, we give intermittent stimulation. So that's another reason why we think perhaps you wouldn't necessarily habituate, because you're getting periods of stimulation versus no stimulation continuously.
Right. So mechanistically, as you were talking about plasticity, so so do you think that that's maybe kind of something that is short term and it's again depotentiating or something? That hence there's no habituation?
So there's an entrainment effect, which would be that you're getting populations of neurons to fire in phase with one another, and that may have benefits. But there's also this clinical after effect that over the course of daily treatments we're seeing effects that outlast the stimulation, and that's different for the people who've got sham stimulation. So that suggests there is some sort of longer term change that outlasts the period of stimulation. And obviously one thing about the entrainment is if you're getting populations of neurons to fire together at the same time, then there's a good chance that you're modifying plasticity.
Hi, yeah. A couple of questions. One is about the effect you've got on stimulating on one wrist and then a lateralized effect in the brain, clearly on the oscillatory stuff that you showed with the MEG. Do you see a lateralized effect on the tics at all?
No, so that's an interesting question. So the point is stimulating the median nerve, which is obviously just these fingers, and yet we're seeing changes in vocal tics, changes in face, which is obviously bilaterally represented, changes in shoulders. So essentially, although we're stimulating very precisely a nerve that has a particular anatomical focus in the brain, we're actually seeing more widespread modulation of behaviour, and as you saw in the fMRI data, we're modulating the whole network.
Yeah, sure. And but it looked like the sort of almost like the whole of the sensory motor cortex was changing its entrainment. And the other question was about, are there any effects on other sensory motor like ability or behaviour while the thing is on?
No, so one thing we checked very early on in probably one of our first papers on this, the first clinical paper we had, was whether or not it interfered with voluntary movements, and the answer is no. Well, it does. I tell a lie. It has something like a 2 millisecond effect on the sort of slowing of movements. So it doesn't have any practical significance on your ability to execute voluntary movements. And then the other thing we checked was whether it has a sort of cognitive effect, whether it sort of distracts you in, say, an attention task, and the answer to that is no. So it seems to primarily modulate these unwanted movements, which some people would say are largely involuntary.
Question at the front here.
Thank you. It was really interesting. My question is, how did you come up with the 10 hertz frequency? Did you study that?
No, so we have explored other. I mean, we've explored beta stimulation as well, and that's effective as well. But we initially started off with mu band frequencies, and it's just as effective at 12 hertz as 10 hertz. So it's fairly arbitrary. And in fact, one thing that we're doing is we're essentially delivering open loop stimulation, and I would suggest now that it could be improved by moving to some sort of closed loop where we're actually looking at what the intrinsic frequency is for that individual and then stimulate it. So in terms of the number of responders, it's probably the case that we could enhance the efficacy and reduce the variability of response by actually moving to some sort of closed loop.
Yeah, that's what I was thinking. Maybe you can titrate it to the particular individual.
Yeah, no, absolutely.
Yeah, because in a sham group, there were still some responders, like 30% or so, with half the frequency. So that's what I was thinking, maybe.
Yes, and I have to come clean here. In subsequent work with EEG, we actually looked at this. So we asked the question, how far below threshold does the stimulation stop being effective? So as soon as you're below threshold, when you stimulate, you don't get a muscle twitch. So you're not getting any sort of efferent copy feedback.
Yeah.
But the question was, are you still getting some sort of physiological response below threshold? And actually what we found is that if you go down to 50%, in many people there is still a response. Even though they can't feel it and whatever, you're still seeing modulation of the EEG. So it turns out our sham probably isn't
sham
a placebo. it has just, it's a reduced intensity stimulation, and in some people it may be more or less effective. So you'd probably have to go down to sort of no stimulation at all to have a real sham.
Yeah, and what do the patients feel? Is it just like a vibration of phone or something? Like, how, is it?
Well, it's mild electrical stimulation, so they sort of, and the response to it varies. I mean, some people find it mildly uncomfortable, others find it completely fine. Many report that it feels relaxing. Once they've got over the shock of, because if you tell them they're going to get electrical stimulation, there's some sort of anxiety. But once they've had it, many people report that they find it very relaxing, actually. But the overarching thing we see from the patients is a spontaneous report that this tick, that's this urge to tick that's always with them, just completely goes away. And they just respond that, we've had people burst into tears that suddenly this urge that's been with them all their lives has suddenly gone.
Yeah, fantastic. It's so clinically impactful. Thank you very much, Stephen. So in the interest of time, we'll move on to the next talk. There are some more questions in the audience, so please do and come and have a chat to speakers afterwards if we didn't manage to get to your questions. Thanks, Stephen. Okay, so we're moving on for the final talk to my talk. So here I'm going to talk about how we can use neuroscience to better understand what's happening with mental health in a workplace context. Just getting the slides ready. Great, thank you. Okay, so I think it's important to think about these links between our working lives and mental health, because we know from data, but also just from all of our own subjective experiences, that our well-being is very interlinked with our working life experience. And there are, of course, many factors at play in that relationship, but one factor that I want to focus on here today is how the amount of time that we spend at work interacts with our well-being. And if you think about a spectrum of how much time we spend at work, we know that extreme ends of that spectrum, whether unemployment or working very long hours, impacts on our well-being. So today I'm going to show you some latest updates. If the slides aren't advancing. Great, okay. I'm going to show you some updates from 2 programmes of work that are ongoing in the lab, but already we've got some really exciting findings to share. So the first of these is looking to characterise the associations between working long hours and physical and mental health in the UK Biobank. And the second is looking to understand the mechanisms behind why we might see improvements in workplace interventions, and specifically a 4-day working week intervention. So first I'm going to show some interim findings from our biobank analyses. Who here has heard of the biobank? Just hands up if you've heard of this. Okay, so almost everyone in the audience, but there are a handful of people for whom this might be a new resource. So this is a mass-scale epidemiological study, studying over 500,000 UK individuals with lots of physical health data, including MRI brain scans. That includes task fMRI, resting state fMRI, T1, T2 structurals, and diffusion MRI as well. And helpfully for us, there is a little bit of employment data in the biobank, including the variable length of working week. So this is how much time are people spending at work. What I'm showing there is the distribution. You can see that a large proportion of participants are working what we would think of as full-time hours in the UK, around 37 or so hours. You can see there are also quite a lot of people working part-time, but also a fair number of people who are working longer than what we might consider to be full-time, of 37 to 40 hours. To tease apart these links between how much time we're spending at work, mental health and brain function, we've taken a multi-pronged approach. So we're looking at these variables in discrete groups of these brackets of how much time people are spending at work, but also looking in a continuous fashion. And that's so that we can understand, is there a linear relationship, or is there a step change where you might cross a threshold in terms of the amount of time you're spending at work, and does mental health suddenly take a turn for the worse? As you can imagine, there are probably a number of covariates associated with working long hours that one might want to consider controlling for in statistical analyses. And so to approach this, we first drew up a list of likely covariates according to the literature and common sense, and then we ran some machine learning, a random forest classifier, to confirm which of those covariates should we take forward to statistical analyses. This is one example where you can see there are a couple identified as unimportant that we were able to eliminate. So firstly, looking at mental health, what we can see is that the probability of feelings of miserableness increases by 0.4%, 0.5%, for every extra hour worked per week. That's shown on the left. And then on the right there, you can see the probability of feelings of nervousness increases by 1%, that's the odds ratio of 0.01 there, for every additional hour worked per week. So this is showing a small but potentially meaningful impact and link between working longer hours and feeling less happy. Next, are there biomarkers of this in the brain? So we focused so far on the resting state data, and what we're looking at is how intrinsic connectivity, how within network connectivity might be associated with working hours. So for example, we're looking at the default mode network shown on the bottom there. So the question here is there a feature in the default mode network associated with length of working week? What we're showing here in green, these are networks that show significant results in terms of an association with working hours. So on the surface, this might suggest that perhaps there are brain bases of longer working weeks. However, when we start to look at this data in a little bit more detail, plotting some of these associations out. Here we're looking at the default mode network just as one example. You can start to see how with very big data you can perhaps get a quite significant result in terms of a p-value, but it may not be terribly meaningful in terms of an actual effect size. So our results so far are suggesting perhaps there is a small but possibly meaningful link there with working hours and mental health, but we're not yet seeing strong evidence that that necessarily is underpinned by meaningfully different brain function, just on what we've checked so far, though, of course, lots more analyses still to do. And if anyone is working with big data or interested in big data, a really useful review article here from Tom Nichols and Steve Smith on the fact that you can often see small effect sizes, significant models, once you move to these really big data sets. Nick Suter, who is our postdoc in the lab leading on the biobank fMRI analysis, had a poster here yesterday. If you didn't manage to catch Nick, we'd love to hear your thoughts on this. So Nick is here, so do feel free to stay and have a chat with us afterwards. So now moving on to think about the 4-day working week. Here I've talked about the impact of working very long hours on mental health, but that's not to say that there isn't still scope for improvement in people who are working more normal full-time hours. And this is what the 4-day working week intervention is looking to explore. So first off, just to define what we mean by that phrase, the 4-day week, because there are a few different definitions. The first bracket is what we call working time reduction. This is where full-time staff reduce the amount of time that they spend at work to 4 days, but they keep their original full-time salary. And often the business is looking to try and maintain outputs, and so the staff need to find efficiencies to get more done in less time. This is sometimes described and thought of as a 100-80-100 model, 100% of the performance in 80% of the time for 100% of the salary. This contrasts with compressed hours. This is where you do the same total number of hours, but you distribute them differently throughout the week. So perhaps you might move to working 4 very long days. And that contrasts again with part-time working, where you reduce the time that you spend at work, but you also reduce your salary. And the data that I'm going to show you here today, the bracket that we've been studying, is that first category of the working time reduction group. And as a neuroscientist, I wanted to study this for 2 reasons. One was because when I saw the media headlines about better well-being, but also better workplace performance, I thought, well, what is it that's driving that? Ultimately, it's the human brain. What is it that produces our work? Whether you're a bricklayer or a marketing manager, that work is being produced by the human brain. And so I thought that the brain is ultimately the explanatory mechanism behind those media headlines that we see about the 4-day week. I also wanted to hopefully do some good knowledge exchange work in helping policymakers by gathering some more objective data to complement some of the self-report scales that we and the others have been taking on the 4-day week. So that has meant it's become a really multidisciplinary project. We have an occupational psychologist on the team looking at helping us with the battery to measure workplace performance. We're taking sleep watch actigraphy and, of course, MRI brain scans as well. So this is the trial protocol and what we ask participants to do when. It's quite involved with a lot of data gathering over quite a period of time. So we start with a couple of baseline weeks. This is when staff are still on their original working patterns, and then they switch to the 4-day week and continue that for a period of 12 weeks. And over that time, we take a short weekly questionnaire battery. We also take a time use diary. This is to ask what do they get up to on their newly free 5th day. The sleep watch I've mentioned. And then for those staff who are up for coming and visiting us to the University of Sussex campus, we're taking a blood sample to look at inflammatory cytokines, an imaging protocol, the details of which I'll show you in a few slides, and some cardiac interoception tasks to look at whether perception of bodily stress signals might be changing on the 4-day week as well. Data collection is still ongoing. We recently got a grant that means we're going for a larger sample size to diversify and look at a wider range of businesses and stratify the sample. But I'm going to show some interim results from the data that we've collected so far, from the questionnaires, some of our sleep diaries and some of the imaging. So first off, looking at our weekly questionnaires, this is the positive and negative affect scale, and what we can see is that as soon as staff switch to the 4-day week, they start to feel better in terms of frequency of negative emotions coming down and positive emotions going up, but it gets progressively better the longer that you've been on the new working schedule. Next, looking at sleep problems, this shows a similar trajectory. As soon as people switch, life starts to improve and sleep starts to get better, but it gets progressively better over time. And we think one reason for that improvement in sleep is the increase that we see in psychological detachment. That's how easy do you find it to switch off from work at the end of the working day, or have you still got your to-do list running through your mind as you're drifting off to sleep? Next, burnout. This is from the Maslach burnout inventory, which specifically measures work-related burnout. Similar pattern, as soon as people switch, they start to feel less burnt out, but as they bank the benefit of more time on the 4-day week, they feel progressively less burnt out. On the flip side, we see a slight but steady climb over time in work engagement. This is how enthusiastic and motivated you feel about work, the sort of feeling you've woken up in the morning and you think, oh, actually, I'm looking forward to going to work today. Oh, I've got the Bacon Symposium to look forward to. Then this is the final measure I'm going to show you from the weekly questionnaires. This is one approach that we've taken to measuring productivity. There's no one gold standard way to do this, and there are pros and cons of different approaches. I'm very happy to chat in the Q&A if that's of interest. But this is one measure that we've used because it can usefully cut across all sorts of different job roles. We've got people from all sorts of different sectors taking part in the study, and this is something that anyone can do in relation to whatever nature of the work they're doing. So we asked them to set themselves 5 work goals or tasks for the coming week ahead at the start of each week, and then at the end of the week to tell us how well they feel they met their 5 goals. So what we see is it takes a few weeks for new routines to bed in. It is very much an experiment for the staff as well, where they have to tweak and iterate and try out new approaches to getting more done in less time. But as time goes on, this steadily climbs. And overall, it's an increase of about 8% in the 4-day week period compared to the baseline period. So why is that? Why do we see an enhancement in performance at work? I think for probably quite a number of reasons, but I think there are 2 psychological routes to enhance performance on a 4-day week, which myself and my occupational psychologist colleague Emma Russell explored in this recent review article. And I'm going to focus in particular on this first proposed route, which suggests that staff being better rested on the 4-day week means that then when they do come back into work, their brain is really firing on all cylinders and they're able to perform at their best. So what's some of the evidence for this? Here, this is some data from our sleep diaries. We haven't analysed the actigraphy yet, but alongside the watches, we asked staff to keep a sleep diary for us. And what we can see is that they don't report changing how much time they're spending in bed, but of the time that they're in bed, they are asleep for 13 minutes longer after switching to the 4-day week. And that's what leads to that increase in sleep efficiency that you can see on the bottom left there. So that's the ratio between the amount of time that people spend in bed and then of that time, how much time they actually asleep for. And this is mirrored by an improvement in sleep quality and with a reduction in sleep problems according to the Pittsburgh Sleep Quality Index. So this is interesting because it's suggesting it's not just changing what's going on at work for people, it's changing what's going on outside of work as well. And that multi-dimensional improvement in well-being seems to be linked to then what goes on in the workplace. So what we've done here is we've looked at how much did someone's well-being change when they switched to the 4-day week and how much did their productivity change. And what we can see is that the more your positive mood goes up, the more your burnout goes down, and the more your sleep improves, the more your productivity goes up. And if you look at the R values there, you can see this is particularly strong for sleep. Next, I'm just replacing goal attainment, productivity with work engagement. And here again, we see a strong relationship with the more that someone's well-being improves, the more their work engagement goes up. Here, the results are even stronger than with productivity. And you can see it's a positive mood boost and a reduction in burnout that's particularly important for that increased enthusiasm and motivation when coming back into the office. So that's some evidence for an association. Of course, it's not causal data, but it's our first indication that there may be a link here between enhancing rest and restoring how people are feeling for when they come back into work. And what's mediating that physiologically? Well, likely some of that is due to what's going on inside the brain. And so we've been taking MRI scans at baseline, and we actually take it on their newly free 5th day on the week that they switch to the 4-day week, so that we're not encroaching on work time too much for research. And then they continue with the 4-day week for another 12 weeks, and then we scan them again to see what's changed. We're taking the Human Connectome project multimodal MRI scan, so that involves a T1 and a T2. We're taking a stop signal task to look at prefrontal function, an interoception task to look at perception of bodily stress signals, resting state fMRI, and also a diffusion MRI scan as well, because we know that sleep, diet and exercise all affect white matter structure, and those aspects of lifestyle that could potentially change on a 4-day week. Here today I'm going to focus on some pilot analyses that we've done on the interoception task and the resting state data. So the interoception task, Chatrin, Camilla and Stephen have very usefully preceded me and introduced what do we mean by that concept interoception. It's the process by which we sense bodily sensations often linked to stress, such as a fast heartbeat, shallow breathing, stomach pains. So the scanner task that we've used is one that's in quite widespread use in the literature, originally implemented by a group in America. In which we ask participants to pay attention to particular sensations they can feel from directed organs, such as the heart, such as the stomach. They're asked to give an intensity rating, and then we have an extraceptive control condition in which they're asked to pay attention to a visual cue instead. And this task gives very reliable activations in the insular cortex when comparing the interoceptive trials to those extraceptive control conditions shown there. So our interim finding from the interoception task suggests that there probably is a brain basis to the improvement in well-being that we see on a 4-day week. So what we see is that mental health symptoms improve after switching to the 4-day week. It's intuitive, but it's nice to see reflected in the data. So symptoms of depression come down, symptoms of anxiety come down, symptoms of stress come down. This is from the DAS-21 depression, anxiety and stress scale. And it seems it's the reduction in anxiety that's particularly coming through in the imaging so far, because what we see is the more that someone's anxiety reduces, the more that their insular cortex activity changes and actually increases after switching to the 4-day week. So tentatively, that might perhaps suggest that after being better rested and less chronically stressed, staff are more accurately able to perceive those bodily sensations of stress and perhaps finding them less intrusive on a day-to-day level. So the final MRI result I'm going to show you is, again, some pilot investigation from our resting state data. Here we were looking at the default mode network, and what we've seen is that the interhemispheric connectivity between the two parietal nodes of the default mode network seems to increase after switching to the 4-day week. And of course, the default mode network, we know that this is associated with creativity and with problem solving. This is a network that is particularly pertinent in a workplace context for enabling people when they do come back into work to perform well. So just to sum up where we're up to so far on the 4-day week project, data collection is still ongoing, and we want to get that larger sample size to stratify into subgroups and really identify who is it that shows the best responses. Is it those who perhaps had poorer sleep to begin with, and so there was more room for improvement? That improved brain function, perhaps prefrontal cortex function, and that's what's associated with an increase in productivity. We also want to stratify by job role and by sector. We're also collecting a control group who don't change working patterns, just to be sure that what we're seeing is due to the 4-day week and not due, for example, to practice effects or Hawthorne effects of being observed. But already we can see that the intervention is doing something for people. They feel better and they seem to be getting on better at work. And there are some early hints that there's a biological basis to that improved performance and to those changes in how people are feeling in terms of well-being. Perhaps a nexus for that might be the insular cortex in particular. Now, I wanted to focus here today on that first route to enhance performance of rest, but I'm just going to touch briefly before we finish on this potential second route to enhance performance on the 4-day week, which is that staff are generally pretty motivated to get their work done in less time in order to retain the benefit of the 4-day week. And that means that they need to engage in behaviour change to find efficiencies. And we have studied some of the ways in which employers go about this. Some of it is sector specific according to the nature of the work, but there are some common threads that run through all the employers we've studied, regardless of the nature of the work. Everyone usually has some sort of desk time or admin time. And so what we've observed is that a lot of employers look carefully at how they schedule the working day. Employees become very intentional about the tasks that they carry out, looking to minimise meetings, minimise distractions. So if you are interested in what you can learn from this for your own working lives and working practices, then please feel free to ask me the questions or come and chat to us afterwards. So I'd like to finish with some thank yous to academic colleagues, in particular to Rhiannon, Nick and Amelia, our 4-day week and biobank team. I'd like to thank the employers who've taken part. These are the employers whose data I've shown you today. Thank you to UKRI for funding my Future Leaders Fellowship and the biobank for enabling us to access the resource, and looking forward to questions. Thank you.
Thank you so much, Charlotte. We have some very quick questions in the front, very fast questions. Oh.
Hello, thank you very much for your talk. I was just curious whether you controlled for working from home after work or on the 5th day or on the weekend, because they might just compensate.
Mm-hmm. Yes, we have actually measured that. I didn't show it in the interest of time, but we do ask them, did you do any work on your newly free 5th day? And we can see some really interesting individual differences coming out there where for the managers and the business owners, they tend to find it harder and have to more strongly resist tapping into work, particularly if there's working from home and hybrid work. So we do see that the business owners and some of the managers might do a little bit of work on that 5th day. So we ask them on a sliding scale. So they're reporting doing maybe 1 or 2 hours of work typically, whereas the general staff members tend to stick to it. But this is an individual difference factor that we'll look to control for in some of the analyses, or potentially even be a variable of interest to consider and investigate. Thank you.
Had a couple of questions in the front.
Thank you for such an exciting talk and such a cool line of research. I was wondering how you're grappling with placebo effects, because they must be enormous.
Mm-hmm. Yeah, and it's impossible to blind. You know you have it or not. And so we're trying to tackle this in a number of different ways. One is by introducing a control group. A second is by taking some control questionnaire measures that we don't expect a 4-day week to change, because they're generally static features of the organisation or personality characteristics. Another is that some of the measures are probably going to be more affected by placebo effects than others, in that it's easy to modify your responses consciously or unconsciously on questionnaires, but slightly harder to modify your responses on blood samples to look at inflammatory cytokines.
I wonder if you thought about maybe doing a manipulation in the control group, because to me it seems like the positive expectations of moving to a 4-day week are incalculable, and you basically, the problem is people are going to go into it with these huge expectations, which will actually affect even things like inflammatory markers in the blood. But I wonder, like, having had a 4-day week imposed on me in the past, where it was just that I couldn't work because I had to do childcare on one of the days, actually I didn't see any of those positive benefits. So I wonder if you could actually have a narrative that would make half your sample have a 4-day week, but maybe not for as positive a reason as they might infer as in your study.
Thanks, Camilla. It's a really valuable point. I think there are two ways in which we're looking to tap into that. And one is that in the control group, we with the 4-day week employers, we do productivity training. That slide that I showed around focus time, minimising distractions, we're doing that with the control employers as well. And so they're getting a sort of productivity intervention, a little bit like Chatran's, active control condition in the autism study. And the second way that we're approaching that is, my mind's gone blank, sorry.
So thanks, that was both fascinating and really important work. I have a question though. So one of the things you talked about subtyping, it strikes me that the type of job you do may have a strong impact on your sense of well-being. And one of the things that strikes me as important is sort of how much control you have over what you're actually doing. So, if you're an Amazon delivery driver, it's not built into your time to even stop and have a wee, then you probably are more stressed than an academic who spends lots of time working from home and thinking or something. So here's sort of a question I have. One of your last slides suggests that employers, when you move to a 4-day week, want to have more structure and control over what you're actually doing. And in some places that may be sort of monitoring your keyboard activity as to how much time. And I'm wondering if that's the case, whether there really would be a benefit. Because I would have thought one of the benefits of, say, working from home or with a 4-day week is that you have a bit more time to structure what you're doing, even if your productivity is going up.
Yeah, so much to unpack there, and you've usefully triggered for me, Stephen, the second part of my answers to Camilla's question around subtypes. So we are taking some demographic information, and also the time use diary will tell us how people are spending their newly free 5th day, and from that we can tell if they're doing childcare, and then be able to stratify in that way. When it comes to autonomy, there are a lot of other questionnaire measures that we took, which I didn't show in the interest of time, and one of those is a measure of how much autonomy do you feel you have over carrying out your job. So this can be a moderating, mediating, or nuisance character control fault, according to how we want to deal with that in the research question. My subjective experience is that generally, fundamentally, in terms of job role autonomy, doesn't seem to change much. That seems to be pretty static. But I've mentioned about diversity, but quite a lot of the employers we've worked with have been in the knowledge economy setting, where there's already a relative, rather than the Amazon drivers, who may need it more, ironically.
I was just wondering if, you present all this data to perhaps employers and they think, yes, this is definitely going to work. But if at the end of that they then exert more control over exactly what people are doing, then it may be counterproductive.
Yeah, generally those we've worked with tend to be really, I didn't show in the interest of time the features of those we've worked with. This is largely being driven by really small employers in the private sector at the moment, who are generally not the ones doing the monitoring of the mouse jiggling. So you sometimes hear about in these big corporates all this sort of monitoring that's going on. Generally there's a lot of communal buy-in from staff who know the business owner well, and there's this sort of pitching in of enabling everyone to do well, without.
We're now running over time, unfortunately.
Okay.
So I think we'll have to call the questions there, and please do come up and talk to us after if you'd like.
Thank you.